Have you ever wondered how a tiny molecule inside your cells decides your eye color, height, or even how your body functions? The answer lies in something called the genetic code. The genetic code is the set of rules that living cells use to translate information stored in DNA into proteins. Proteins are essential molecules that perform most of the functions in our body. To understand the genetic code, we first need to understand the DNA. So, what is DNA? It is deoxyribonucleic acid. The molecules that carries genetic information in almost all living organisms. And it is often called a blueprint of life. The structure of DNA was discovered by the scientists James Watson and Francis Crick using crucial data from Rosalind Franklin. DNA has a double helix structure and is made of smaller units called nucleotides. Each nucleotide contains one of four nitrogenous bases: adenine, thymine, cytosine, and guanine. These bases pair specifically. Adenine pairs with thymine and cytosine pairs with guanine. The sequence of these bases forms the instructions that cells use to build proteins. The flow of genetic information in a cell follows a concept called central dogma, which is proposed by Francis Crick. The central dogma explains that genetic informations flow in three main steps: DNA to RNA, then RNA to protein. First, DNA is copied into messenger RNA in a process called transcription. Then the messenger RNA moves to a structure called the ribosome, where it is translated into proteins. This process is called translation. This is where the genetic code becomes important. Then what is the genetic code? It is the system that determines how the sequence of nucleotides in a messenger RNA is converted into amino acids. Amino acids are the building blocks of proteins. The code is read in groups of 3 nucleotides called codons. Each codon specifies a particular amino acid. For example, the codon AUG codes for the amino acid methionine, which also act as the start signal for protein synthesis. There are 4 bases in RNA: adenine, uracil, cytosine, and guanine. Since codons are made of 3 bases, there are 4 raised to 3 or 64 possible codons. Out of these, 61 codons codes for the amino acids. 3 codons act as stop signals. The stop codons are UAA, UAG, and UGA. These signals tell the ribosome when to stop building the protein. Then, now we check what are the properties of the genetic code. It has several important characteristics. First of all, it is a triplet code. That means each codon consists of 3 nucleotides. Second one is, it is degenerate, which means that multiple codons can code for the same amino acid. Third one is, the genetic code is universal. Nearly all the organisms from bacteria to humans use the same genetic code. The fourth one is, it is non-overlapping. That means each nucleotide belongs to only one codon. And finally, it is comaless, that is, codons are read continuously without gaps. And the genetic code was discovered through experiments by scientists such as Marshall Nirenberg and Hargobind Khorana. Now, coming to the importance of genetic code. The genetic code is essential for life. It controls how cells produce proteins that perform functions such as metabolism, growth, and repair. Understanding the genetic code has also allowed scientists to develop modern technologies like genetic engineering and gene editing. And one powerful gene editing technology is called CRISPR, which allows the scientists to modify DNA with great precision.